All right, we have been talking about glycogen. We talked last class about its synthesis. You will recall that the process utilizes UDP glucose, so the substrate for both glycogen and glycogen synthase is UDP glucose. Glycogen is used as an anchor, a starting point, a template for the production of glycogen, and approximately 7 or 8 glucose molecules are added to a tyrosine present on the glycogen. Thereafter, glycogen synthase takes over and extends the polymer to great lengths. There is also a branching enzyme that introduces these branch points about every 8 to 12 glucose molecules. Today we're going to talk about glycogen breakdown, and I'll be using slides to talk about that, and we'll also talk about, or begin talking about, the regulation of glycogen, both glycogen synthesis and breakdown. And let me just preface this, regulation by saying that the regulation of glycogen synthesis and breakdown is under hormonal control. So there are really, with glycolysis, there were many allosteric factors that governed whether we were doing, whether we were increasing one enzyme's activity or reducing it. But with glycogen, it is really hormonal control. So with that as sort of an intro, I am going to move to slides. So glycogen breakdown is initially is accomplished by an enzyme called glycogen phosphorylase. Sometimes it is referred to as phosphorylase A and sometimes phosphorylase B, depending on whether it is activated or inactive. But we are, I'll refer to it as glycogen phosphorylase just to help keep things clearer. All right, in this process, this glycogen phosphorylase utilizes a critical cofactor. By cofactor, I mean this is a molecule that is independent. It's not a protein, but it is critical for the activity of the glycogen phosphorylase. This particular factor is called pyridoxal phosphate. We'll see it in a minute, and we'll talk a little bit about its involvement in the reaction. But it is going to activate a phosphate, an inorganic phosphate, which is going to be the attacking species. It's going to attack the 1 carbon of the terminal glucose in a polymer of glycogen. I think I mentioned last time that you can have multiple copies of the glycogen phosphorylase on the surface of a glycogen particle. Remember, it has all of these branches which end up producing all of these endpoints, and those can be acted upon independently by multiple copies of glycogen phosphorylase. All right. So this is the pyridoxal phosphate molecule. It is a vitamin. That is to say, our body does not produce this particular molecule. We rely on this coming to us from dietary sources. It is going to, so it has a aldehyde group, as you can see here, that is going to bind to the enzyme. Okay? So that's going to bind to a side group, presumably a lysine, on the enzyme. But it is this phosphate out on this end of it that is going to activate an inorganic phosphate. So this pyridoxal phosphate will be at the active site of this of the enzyme. This phosphate will be positioned next to an inorganic phosphate that is going to be bound by the enzyme. And the result is that that activated phosphate is going to come in. Let's see, get back here again. It's going to come in and attack. Sorry. It's really inconvenient here. They have no room for the mouse. This one carbon of the terminal glucose of a branch of the glycogen. So the phosphate comes in, the negative charge is going to attack that carbonyl carbon. Remember, it was an aldehyde to begin with. It now is this acetal group, but it will come in and attack. That's going to result in the production of glucose 1-phosphate. This oxygen here that is left behind, this oxygen left here will pick up a proton from solution, presumably, and will now have, continue to have glycogen, but it will be one glucose molecule shorter. And this process can be repeated until we get in the within 4 glucose molecules of a branch point. Okay, so this is the basic glycogen breakdown process. Glycogen phosphorylase, by using pyridoxal phosphate, activates a phosphate, attacks the 1 carbon of the terminal glucose molecule, releasing glucose 1-phosphate. All right. So this process proceeds until we're within 4 glucose molecules of a branch point. And at that point, the glycogen molecule will no longer fit in the glycogen phosphorylase active site. So rather than having everything stall at that point, the body has another enzyme called the debranching enzyme, which describes well its activity, which is going to come in and move 3 out of the remaining 4 glucose molecules on the branch, and it's going to move them to the main chain. So these 3 molecules then end up coming down and being added now to the main chain of the glycogen. So this is a transferase activity. This same enzyme also has a hydrolase activity where it's going to hydrolyze off the final glucose molecule right here as just glucose. It's not going to produce glucose 1-phosphate. It is going to simply hydrolyze that off. That will be will be phosphorylated by hexokinase thereafter, and that glucose will end up being glucose 6-phosphate. We now have a straight chain again, and so the glycogen phosphorylase will come in and bring about the breakdown of the glycogen until we reach another branch point. And then the debranching enzyme will come in and remove the branch in 2 steps. A transfer step that takes 3 of the 4 glucose molecules and adds it to the main chain and then hydrolyzes off the final glucose. So glucose 1-phosphate, if we are producing that as far as glycogen breakdown, that is not immediately usable for glycolysis. So there is an additional enzyme that is part of glycogen breakdown. It is called the phosphoglucomutase. And it's able to interconvert glucose 1-phosphate to glucose 6-phosphate, or conversely, glucose 6-phosphate to glucose 1-phosphate. So we saw this previously. So in the production of UDP glucose, we initially converted glucose 6-phosphate to glucose 1-phosphate. Then that got coupled to part of the UTP molecule to produce UDP glucose. Again, the enzyme that was able to convert glucose 6-phosphate to glucose 1-phosphate for glycogen synthesis is the same enzyme that is now able to take the glucose 1-phosphate, which is part of glycogen breakdown, and convert it into glucose 6-phosphate, allowing it to enter glycolysis. So this reaction is somewhat analogous to the phosphoglycerate mutase. This is step 8 in glycolysis, in that we are going to add a second phosphate to our glucose 1-phosphate, and then we're going to remove the opposing phosphate, leaving us now with glucose 6-phosphate. This particular enzyme has a modified serine at the active site. This serine contains a phosphate coupled to that hydroxyl group. And that is donated then to produce, is added to produce this glucose 1,6-bisphosphate. And then this same serine is able to remove the 6-phospho group, the 1-phospho group, leaving us now with glucose 6-phosphate. This process goes easily in either direction. But if we are breaking down glycogen, we will have much more of the glucose 1-phosphate, which will drive the reaction towards the formation of glucose 6-phosphate, which will be utilized in glycolysis. All right, are there questions here? Because this pretty much summarizes the glycogen breakdown process. Yes?
I have a couple questions, but it's mostly for my own curiosity.
Yeah.
Why 4 glucose molecules? Why does it stop at 4?
Okay, the question is why with the glycogen phosphorylase does it stop removing glucoses when it's 4 sugars away from the branch point? And I think the simple answer is at that point this branch tends to not fit in the active site of the glycogen phosphorylase. So if you can imagine, this branch is cumbersome, if you will. It creates this sort of problem to be able to actually fit the substrate into the active site of glycogen phosphorylase. Had the glycogen phosphorylase been designed differently, it might have happened at 5 sugars, or it might have happened at 3 sugars. It just so happens that once you get to that close to a branch point, it no longer fits in the active site. So, yeah.
And then which substances have a pyridoxal phosphate? Like you said, there's a vitamin,
Which types of food have pyridoxal phosphate? It's one of the B vitamins. I forget which one it is. It's like B6 or something like that. I can't remember. Do we have any dietary people here? Anyway, it is a common vitamin, and I think it's present in many different kinds of foods. I don't think it's particularly rare. So, but I don't specifically know which foods are high in pyridoxal phosphate. All right, one further consideration in terms of glycogen breakdown in the liver, and that is being able to export it to the circulation. So glycogen is stored in the liver, and glycogen, well, is formed and stored in the liver. It's formed and stored in skeletal muscle. But skeletal muscle does not have the ability to export glucose. So the glycogen is used then just locally in skeletal muscle. However, the liver has the capacity to release the glucose into the circulation, and in doing so, sustaining critical tissues. These tissues, at least in the short term, that is, if you experience hypoglycemia, those tissues are the brain, the heart, and the kidney. If we lose any one of these, we are unable to survive. Okay. Now, it turns out that the export of glucose ends up being a very complicated process, as diagrammed in this slide. We will break things down in the cytosol. We'll break things down in the cytosol for glycogen or for carrying out gluconeogenesis. Most of that takes place in the cytosol. The first step doesn't, but most of it does. And in either case, whether we're doing gluconeogenesis or glycogen breakdown, we end up with glucose 6-phosphate, and it's present in the cytosol. To be processed further to glucose requires that it enter the endoplasmic reticulum. This is a specialized compartment within the cell, an organelle. And to get it there, it requires a specialized transporter. This is this glucose 6-phosphate transporter, or T1, which allows glucose 6-phosphate to move through the membrane of the endoplasmic reticulum into its lumen. There is then the final enzyme in this, either in the gluconeogenic process or the final enzyme necessary to produce glucose, that is this glucose 6-phosphatase, which removes the phosphate. We now have glucose, but it's present in the ER, and to get it out of the ER, we need yet another transporter, this glucose transporter T2, which is present in the membrane of the endoplasmic reticulum. We now have glucose present in the cytosol, and it can exit the cell through the GLUT2 transporter. As mentioned previously, the GLUT2 transporter is found in the liver cells, hepatocytes, in the pancreatic beta cells, but because of its being here in a permanent way, it's not insulin dependent, the glucose then can move out of the cell. So it really depends on concentration, but if we're generating glucose in the cell, either through gluconeogenesis or glycogen breakdown, the concentration in the cell will be greater than the concentration in the circulation, and the glucose will move out of the cell. It is necessary that the hexokinase 4 glucokinase not be active, okay? So at modest, low levels of glucose as produced by glycogen breakdown or gluconeogenesis, we have to assume that there's not enough to activate it, not enough to lead to its relocation, translocation from the nucleus to the cytosol. Otherwise, it would simply convert the glucose back to glucose 6-phosphate and preclude glucose from being present in the cytosol for export. Okay, so a complicated process to get it out of the cell and into the circulation. You'll also recall that under conditions of hypoglycemia, the cortisol goes up, and that leads to insulin resistance in most peripheral tissues. It leads to a marked reduction in the use of glucose by other tissues that are non-critical. So in that sense, the glucose produced and released from the liver is free to make its way to those tissues where it is needed for survival. Okay, questions about this? All right, we are going to now move on and begin a discussion of glycogen regulation. And I will say that it is pretty complicated. So, as you will, oops, let's see what happened here. Is it sharing?
Yeah.
All right, so immediately one is impressed with just how complicated this whole business is. As I mentioned, so the ruling, the overarching principle is that glycogen regulation is under hormonal control, and it is regulated, the enzymes involved and so forth are regulated by phosphorylation or dephosphorylation. So hormones then lead to phosphorylation or dephosphorylation of critical enzymes that participate in either glycogen synthesis or glycogen breakdown. So this slide talks about glycogen phosphorylase. Glycogen phosphorylase is the enzyme that is responsible for the release of glucose, glucose 1-phosphate, from glycogen. So up here at the top, we find these two hormones that are going to govern the activation of glycogen phosphorylase. Glucagon is going to regulate the liver. It has a receptor in the cell membrane of hepatocytes. As it binds there, it binds to a receptor. This is going to lead to the activation of protein kinase A. In the skeletal muscle, epinephrine does the same thing. It binds to its cell surface receptor, leads to the activation of protein kinase A. Protein kinase A, in turn, is going to phosphorylate an enzyme called phosphorylase kinase, or maybe more descriptively, glycogen phosphorylase kinase. This enzyme is going to be responsible for phosphorylating in activating glycogen phosphorylase. So here's this phosphorylase kinase. It is active when it is phosphorylated. The pKa is phosphorylating it, activating it, and in turn, it is going to phosphorylate and activate glycogen phosphorylase. Okay. All right, so glycogen phosphorylase, when activated, then breaks down glycogen, releasing glucose 1-phosphate, which is used then to for those purposes just described. I think part of showing this slide in this way is to give you the sense that there is a multiplication factor, an amplification factor as we go through this process. So glucagon, let's say a single copy of glucagon combined to its receptor, it activates this adenylate cyclase activity, which is going to activate pKa. Well, maybe it's going to produce 10 copies of active pKa. And the pKa as an enzyme can process multiple substrates, so it can process multiple copies of the phosphorylase kinase. And so now you get another amplification. So maybe now you have 100 copies of the phosphorylase kinase that are active. And they in turn, as enzymes, can process multiple copies of the glycogen phosphorylase, which can lead then to, maybe 1,000 copies of glycogen phosphorylase being activated, and those can be distributed around the glycogen particle and lead to the release of glucose 1-phosphate, converted into glucose 6-phosphate onto glucose in the liver. In other words, it brings about a very rapid mobilization of glucose from glycogen. So we get the benefit of this in a matter of a few minutes. So let's just say that you go out running. You may get by for 10, 15, 20 minutes on available available glucose in your circulation. But after about 10 or 15 minutes, then you tap into glycogen stores, unless you're on the keto diet, which means you have no glycogen and you have no endurance. But once you tap into the glycogen, then you begin to resupply glucose. You're able to continue. In the skeletal muscle, that glycogen is used through glycolysis to produce more ATP, maintain activity, and this can go on for quite a while. So if you are a conditioned runner, your glycogen stores, if you've eaten quite a bit of carbohydrate in the days leading up to your race, may allow you to run for 3 hours on glycogen. But then you run out of glycogen. This is called hitting the wall. And it feels like you've hit a wall. I mean, physically, you feel awful. And it represents this transition from no glycogen to trying to use other sources of energy, ketone bodies, fatty acids, and so forth, to maintain your effort. And for well-trained athletes, this transition goes pretty smoothly. But for the person who doesn't run marathons routinely, it is a very unpleasant event. Okay, so this is glycogen breakdown, activated by glucagon in the liver, epinephrine in the skeletal muscle. The process involves phosphorylation by activation of pKa, phosphorylation of this enzyme phosphorylase kinase, which is going to phosphorylate and activate on glycogen phosphorylase, leading to glycogen breakdown. This enzyme is coordinately regulated, so it is activated by glucagon or epinephrine, but it is going to be inhibited by, any guesses? It's going to be inactivated by insulin. Okay, so remember we have these two major players, glucagon and insulin. So if we have abundant glucose or carbohydrate, insulin levels go up. At that point, you don't want to be breaking down glycogen. If anything, you want to synthesize glycogen. And so to accomplish that, the insulin leads to the activation of this phosphoprotein phosphatase 1. Okay, we saw this enzyme previously with that bifunctional enzyme that produces fructose 2,6-bisphosphate or removes fructose 2,6-bisphosphate. We saw it with hepatic form of pyruvate kinase, but here it is again. Insulin leads to the, activates this PP1, which dephosphorylates glycogen phosphorylase and inactivates it. So on the one hand, we have glucagon leading to its phosphorylation, leading to its activation, leading to glycogen breakdown. Remember, glucagon is a response to hypoglycemia, so that makes sense. If we have adequate, abundant carbohydrate in the diet, then we want to stop the breakdown of glycogen and produce more glycogen. And so this process then is coordinately regulated by these different hormones. One sort of side note about the glycogen phosphorylase is that there is actually an allosteric binding site for glucose on the glycogen phosphorylase. And when glucose levels are high within the cell, it leads to a conformational change which exposes this phosphorylated serine and facilitates the removal of the phosphate inactivating glycogen phosphorylase. Okay, so again, we have this serine up here. When it is phosphorylated, we have active glycogen phosphorylase. That is promoted by glucagon through protein kinase A or epinephrine through protein kinase A. Conversely, if we have high glucose, we're going to activate this phosphoprotein phosphatase 1, leading to its dephosphorylation and inactivation. Okay. The glycogen synthase is, its regulation is a bit more complicated. Its activation is actually fairly straightforward. So if you're thinking about this, when would I want to activate glycogen synthase? When I'm hypoglycemic or when I have adequate, abundant carbohydrate? Yeah, so if I have sugar to work with, let's synthesize some glycogen. So this is going to be under, the activation is going to be under insulin control. So here comes insulin again. It leads to the increased levels of this phosphoprotein phosphatase 1, right here. And what it does is it's going to remove a series of phosphates from glycogen synthase, and that leads to the activation of glycogen synthase, leading to increased production of glycogen. Okay, that part is fairly straightforward. Now, the inactivation of glycogen synthase is more complicated. somewhere out there, it is under glucagon and epinephrine's control, but the exact mechanisms are unknown at this time. Suffice it to say that the glycogen synthase has a series of serines that are 4 amino acids apart. There is an enzyme called casein kinase 2, which is going to come in and add a phosphate to one of these serine molecules. This can be thought of as a priming event. This phosphoserine has to exist before you bring in a second enzyme, this glycogen synthase kinase 3, which is going to then phosphorylate several serine molecules, and in doing so, inactivates the glycogen synthase. Let's look at this in a little bit more detail up close. So this first serine, as shown here, has to be phosphorylated before the GSK3 can bind to glycogen synthase and act on it. And as mentioned, that first phosphate is added by casein kinase 2. This GSK3 is a crazy kind of enzyme. So it has the substrate binding site, which is shown here, and it's surrounded by all of these positively charged groups. So you have the negative phosphate, the 2 negative charges on the phosphate. It leads to the GSK3 binding to the glycogen synthase. But then the catalytic site is next door. It's 4 amino acids further along, or it's a distance equivalent to 4 amino acids further along. And it's going to add a phosphate to a serine 4 amino acids away from that initial serine molecule. And notice that you have this series of serines. You have them here, 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 here. Now, the remarkable thing is that after I phosphorylate this serine right here, in the catalytic site, the whole enzyme is going to move. It's going to move 4 amino acids farther along. This binding site is now going to overlay this newly phosphorylated second serine, and the active site, the catalytic site, is going to be placed over a hydroxyl group on a serine 4 amino acids away. And this continues to add phosphates until 3, 4, 5 copies of serine now have phosphate present on them. This leads to its inactivation. Okay? So, and you'll recall that the phosphoprotein phosphatase 1 under insulin's control is able to remove all of these phosphates and restore glycogen synthase to full activity. But these other 2 enzymes, the casein kinase 2 and the glycogen synthase kinase 3, are going to lead to its inactivation. Okay. Questions? This is very busy stuff. A lot of steps.
I have a question.
Yes.
Most of the time, enzymes are activated when they're phosphorylated. Is that right?
No.
So it just depends on the enzyme?
Yes.
Because in this case, it's inactivated when it's phosphorylated.
Well, the glycogen synthase is inactive when phosphorylated. The glycogen phosphorylase is active when it's phosphorylated. Conversely, glycogen phosphorylase is inactivated by dephosphorylation, whereas glycogen synthase is activated by dephosphorylation. It really depends on the enzyme, and so it's a little bit, you can't make general statements about what phosphorylation does to enzymes generally. It really depends on the enzyme. All right, I think I'm going to end here. There's a little bit more that we'll talk about on Monday related to glycogen regulation, but I wanted to finish this story. So let me ditch the slides here. So I had mentioned that I had gone off to grad school, but just prior to leaving, I had run into a young woman that I'd known previously. I was living in Connecticut at the time, but I continued to come back to Utah to attend my National Guard meetings, which were monthly, as I tried to get into a National Guard or Reserve unit in Connecticut. And as I would go back to Utah, I would call or visit this young woman, and it became apparent that her life seemed to be unraveling. And this was manifest in a couple ways. One, as I talked to her, to begin with, she was so excited about this new lifestyle that she had adopted. She had thrown off all the burdens of, of family and church and these friends who were phony and all this sort of stuff, and was living this completely spontaneous lifestyle. But as time went on, she seemed less and less enthusiastic about her lifestyle. She was less excited, less happy. I noticed that her drug use seemed to be going up. To begin with, she and her friends on the weekend might indulge in marijuana and do this as part of some group activity. But as she described her life, it became apparent that she was using drugs almost daily to keep her spirits up. And my concern was that her drug use was moving from things that were less harmful to things that were more addictive and more problematic. And she started having, she just didn't go out. She used to be very gregarious and outgoing and fun-loving, but over the time that I kept going back to Utah, it became apparent that she really was living a very insular life. She didn't go out. She didn't interact with friends very often. She just stayed in her room and did her things. And this concerned me. The trajectory was down, and it was going down pretty steeply. I didn't have to go back to Utah after a few months, and so I didn't see her until the following summer. I did occasionally get letters from her, but I saw her that following summer after I had gone to grad school. And it alarmed me. She just was miserable, and she just seemed to, her situation just seemed to be very, very scary, frankly. And so I came back to Connecticut, back to New Haven, and really very, very concerned about her. Now, you'll recall that my coming back from Germany, I was not living a religious lifestyle. And while I went back to church when I was in New Haven, I still hadn't completely eliminated some of these problems from my life. So one evening, maybe the day I got back, I don't remember, but I got down on my knees and I pleaded for Julie. I pleaded with my Heavenly Father that He would guide me to know how to help her, or what could be done to improve the situation. And as I was giving this prayer, it was as if somebody came into the room. Suddenly, the room was warm, and I felt surrounded by warmth and love. And I received an answer, and the answer went something like this. Stephen, you can see that things are not going well for Julie. You can see that she's becoming more and more distant and more dependent on drugs, and is really having a very hard time. But in your condition, given that you are not living the commandments, you are not in a position to help her. And that was pretty disappointing. It was true, but I didn't want to hear it. Then I got a second answer, and it went something like this. Stephen, even in your fallen state, you feel concern and love for Julie. You can see where this is going. Even with your own problems, you still have the ability to recognize the distress that she is in. Imagine how I, as a God, feel about you. You are breaking my heart. It was the first time I realized that my behavior, my personal behavior, was registered pain to my Savior. It was a real wake-up call for me, because not only was I unable to help Julie, but I suddenly recognized that my feelings for her were small compared to my Heavenly Father and my Savior's feelings for me and my own deteriorating situation. Anyway, I share that with you in hopes that you know that God loves us, and that when we injure ourselves, it injures Him. Anyway, thank you very much. Enjoy your weekend. Stay safe. Please stay healthy.